All right, so for our next session, we're going to be talking about therapeutic treatment approaches that are now in the clinic and clinical trials. So they've overcome all the preclinical studies and gotten an IND opened with the FDA. We're going to do the same format. Each of our speakers is going to talk for about 10 minutes, and then we'll do questions at the end. So first up, I would like to welcome Nancy Ruiz from Larimar Therapeutics.
Good morning, everyone. First, I want to thank all of the previous speakers because they've kind of set the tone for what I'm going to talk about. I want to walk you through our journey with CTI-1601, which is a molecule which is being studied as a potential mitochondrial frataxin replacement therapy. I think by now you know the importance of frataxin. Because I'm going to talk about where we're going, I have to present that slide that everybody is presenting. You could read it at your own leisure. But let's start by looking at how does human frataxin look like. On the left, you will see what mitochondrial targeting sequence, which is in green, and the blue section is the human frataxin. Where you see the three circles, you'll see that is where the molecule is cleaved or broken up so that human frataxin then remains inside the mitochondria. When you look at the right, you'll see that the only difference between the naturally occurring frataxin and CTI-1606 is that cell-penetrating peptide, which actually allows the molecule to enter the cell, then enter the mitochondria, and then get cleaved at the same place, leaving the human frataxin inside the cell. So it's basically very similar to the naturally occurring cell. As you've heard from other speakers, before we actually do human studies, we do animal pharmacology studies. And these help evaluate the potential effect and safety of a product, and it helps evaluate the proof of concept. Does the product have the potential to do what it's intended to do? For that, we use knockout mouse, and a laboratory mouse that is genetically engineered in which one or more genes have been turned off or knocked out. These mice are used to study what happens in an organism when a particular gene is either absent or not functioning appropriately. In our case, we looked at cardiac knockout mouse, and we also looked at a neurologic knockout model. What we demonstrated was that we could, in these animals, deliver frataxin to mitochondria using CTI-1601. So to get to the bottom line, we showed that CTI-1601 prevents the development of ataxic gait in these mice. Furthermore, we also showed, and I'll show you a slide later in the cardiac model, that these mice survived for a longer period of time. And we also showed that the human frataxin was found in the brain, in the dorsal root ganglia, and in the spinal cord. So this was very exciting and gave us a lot of hope. We then looked at the cardiac mouse model, and we found that the median survival of the treated mice was 166 days versus 98 in those that did not receive the drug. 87.5% of the mice actually survived versus 33% of those who received the vehicle. And as I said before, we demonstrated that CTI-1601 is capable of delivering sufficient amounts of frataxin to the mitochondria. We looked at cardiac function, and what you'll see here is the animals in red. The line in red is those that are the knockout mice that did not receive CTI-1601, whereas the line that is blue are the ones that did receive CTI-1601, and you see that the cardiac function is maintained compared to those that did not receive the drug. So what we saw then from the cardiac knockout model is that it extended survival, demonstrated the ability to deliver frataxin to mitochondria. Somebody mentioned succinate dehydrogenase as a marker of mitochondrial function, and we showed that we increased the activity of the mitochondria. And it prevented left ventricular dilatation and maintained cardiac function. From the neurologic knockout mouse, we saw that it prevented the development of ataxic gait, showed that treated mice survived longer than untreated mice, and demonstrated CNS penetration. So that gave us the ability to start our phase 1 clinical program, in which we did a single ascending dose and a multiple ascending dose study in patients with Friedreich's ataxia. In the multiple ascending dose study, we dosed 25 milligrams for 4 days, followed by every 3 days, 50 milligrams for 7 days, followed by every other day, and 100 milligrams for 13 days. And the results, what you'll see here is, if you look at the graph on the right, you'll see the changes from baseline, from when the patients entered the study, and you'll see in purple and in blue, dark blue, that there are increases in the frataxin level as observed in the skin, as observed in the cells from the inside of the mouth, which is a common place where we look at frataxin. And also, although there's a little bit more variability, we saw that it also increased in platelets. So this showed us that in humans, we also were able to deliver frataxin to the cells. Now, I think Jen and others have mentioned that carriers don't have the same amount of frataxin as healthy individuals. However, they don't exhibit symptoms. And what really encouraged us was that from our data, we saw that both at the 50 and the 100 milligram dose, we actually achieved levels, not 50%, but about 60% of those in normal individuals or in unaffected individuals. So again, that was extremely encouraging to us. And in summary then, CTI-1601 appears to be generally well tolerated. As others have mentioned, safety is the main objective of a phase 1 program. The most common adverse events were actually injection site reactions, which were seen in all the patients that received drug, but also in 43% of patients who received placebo. Daily dosing of the drug resulted in dose-dependent increases in frataxin levels from baseline compared to placebo. And I didn't present pharmacokinetics, but the pharmacokinetics supports daily dosing. And more importantly, or most importantly and exciting, daily subcutaneous administration of 50 and 100 milligram doses resulted in frataxin levels in buccal cells that are at or in excess of those we would expect to see in phenotypically normal heterozygous carriers who have frataxin levels of about 50% of unaffected persons. So our next steps then are to continue to interact with the regulatory agency, with the FDA. We will start next year, the first half of next year, our JIVE study, which is an open label extension study for the patients who have participated in our previous studies. Everybody will receive CTI-1601, and that will be a long-term study. We will initiate a study in pediatrics in patients less than 18 years of age at the beginning of next year. And then we will move on to a global double-blind placebo-controlled trial as of now, which we expect to initiate in the first half of 2023. So thank you very much, and I will take questions at the end of the session.
Thank you, Nancy. Next up, we have Mark Madej from Retrotope. We got a lot of clinical programs. This is exciting.
Thank you for having me. David Lynch was just reminding me that we met 40 years ago, I think, on a baseball team back in Baltimore. And I went on to become a cardiologist, not a neurologist. And I'm not exactly sure what his career path was, but if you told me we were going to end up, if you told me that we were going to end up here in this kind of venue, I would have said you were crazy. So I'm going to tell you about a clinical trial that's ongoing right now at Retrotope. Do I advance the slides?
Yes.
Got it. So the top is a chemical formula, a molecular formula for linoleic acid. Linoleic acid is one of the polyunsaturated fatty acids that are essential to your diet. You can't make it, so you have to eat it. But fortunately, it is delicious, and it's almost in everything. Potato chips, any kind of fried food, almost anything has some linoleic acid in it. And the problem with linoleic acid is that it is susceptible to oxidative damage. It makes up most of the peripheral cell membrane. So the skeletal muscle, the cardiac muscle has a lot of linoleic acid in its membranes and in the mitochondrial membranes. And in the brain, it is elongated to form arachidonic acid. So those two red hydrogen molecules that you see making up the backbone are particularly prone to it. And Retrotope has a family of compounds that substitute various susceptible hydrogen molecules. It's substituted with deuterium, which is an isotope of, it's not radioactive, despite what people's preconceptions are, but it is naturally occurring. It happened at the Big Bang, and there's a certain amount of it around, and it's not going to be any more or less. But we substitute them, and it makes that molecule far less, if not immune to oxidative damage. Well, as it happens, the free iron that is not contained in, you can see in the top line, the normal gene produces frataxin. Frataxin cages and limits the amount of free iron inside the cell. And so there's a certain amount of lipid peroxidation that goes on, but your body has mechanisms for dealing with that. In the Friedreich's ataxia disease state, there's not as much frataxin around, or the frataxin activity is reduced. There's a lot more free iron, and free iron is a potent stimulant and promoter of lipid peroxidation, and that, we believe that's part of the pathophysiology of it. After the administration of our drug, RT001, the membranes, despite the increased amount of free iron, the membranes are far less susceptible to lipid peroxidation. And so we are hopeful that it protects against progression and perhaps facilitates improvement. We did a phase 2A study a few years ago in which just after a month of treatment, we were able to show significant change in peak workload in a very small number of patients. And maybe some of the people are in the room that participated in that trial. This is not data from, I don't have data from our current trial yet. I'll tell you about that in a moment. But we just had a trial in a disease called infantile neuroaxonal dystrophy, in which there's another reason for increased lipid peroxidation. And we actually showed a survival benefit in those patients. And so we're newly excited about proof of the mechanism of action working. And also in that disease, we saw a series of trends. Everything's in the right direction. So we're very excited about the fact that we've been able to show a clinical benefit in a group of patients with a disease of neurodegeneration caused by lipid peroxidation. So this is the study, and that we have completed the randomized phase for. I think we enrolled 65 patients total. We had early terminations in 12, and as you might imagine, trying to conduct a clinical trial during COVID has been a challenge, getting people to come back and things. And so most of the early terminations were due to logistical considerations rather than any kind of intolerance. Very few SAEs. We had a DSMB meeting and they gave permission to continue the trial without interruption. None of the SAE, the serious adverse events, were related to the drug. Our first patient was treated January 7th. The last patient started treatment in November 20th of 2020, and we had our last patient last visit last month. So we are in the process of cleaning up the data. That usually takes about a month. We anticipate once all of the data's been entered, locking the database and doing an analysis. That should begin very soon, and we hope to have some data for you shortly. In closing, I wanted to thank the investigators, the coordinators, the nurses. FARA has been unbelievable in supporting us, in helping us get things started, and they're great about coordinating between the sponsors, because you know there's only a finite number of you out there, and we need to maximize the benefits and minimize the risks for patients, and so they've been helpful in facilitating that process. And of course, we want to thank the patients and their families, because we really know what an ordeal this was to try to visit, get tested before coming to the hospital, et cetera. You know what life has been like this past year, but from the looks of this crowd, it looks like there's an end in sight and light at the end of the tunnel. So thank you very much. I'll finish a little bit early.
Just to piggyback a minute on what Mark was saying about the challenges that we have had during the past 2 years conducting clinical trials, all the folks you're hearing from today have worked with our clinicians and worked with all of you to figure out how to do these trials during a pandemic. That was a whole nother added complication that nobody saw coming, for sure. And during that time, we've had more than 350 individuals with FA participate in clinical trials in about 18 months. And not only in the US, but that's the US, North America, South America, Europe, and Australia. And that doesn't include all our non-interventional clinical trials as well. And that really, I'm really glad, Mark, you brought up the point of acknowledging the investigators and the coordinators for working with our sponsors to figure out how to do that and do that as safely as possible for all of our patient families coming. And it's really been remarkable, and it's been a pleasure. I've worked with all of you through this process to figure this out, and it really has been an evolving learning process that we didn't count on when we started planning these trials. So thank you all very much for that. Next up, we have Tracy Schilling from PTC.
Hello, everyone. I'm Tracy Schilling. I'm an adult neurologist with neuromuscular subspecialty training, and I'll be talking about our program looking at vatiquinone, which has gone through a couple of different names through the years. It's also known as PTC 743 and EPI 743. So as I said, I represent PTC Therapeutics. We're a biotechnology company that has been around for a little over 20 years now. We have a really robust pipeline, and we have a high interest in diseases that are rare and with unmet need. You'll see that we have 2 programs in Friedreich's ataxia. One is the PTC 743 vatiquinone program that I'll talk to you about today. We also have an earlier stage gene therapy program in development as well. So we're really committed not only to rare diseases in general, but to Friedreich's ataxia specifically. Jen and our other presenters have done a really great job in talking about some of the pathophysiology of Friedreich's ataxia. What I wanted to highlight is another component of the decrease of frataxin and what that causes that has been shown to be really important in causing the signs and symptoms of Friedreich's ataxia, and that's something called ferroptosis. Ferroptosis is a programmed cell death pathway, and it's completely normal when it's regulated like it's supposed to be. It's actually really helpful if you have cells that are damaged by some other condition. It removes those cells so that things don't get out of control. But in cases where it's not regulated properly, like in Friedrich's ataxia, it can cause death of cells that shouldn't otherwise be removed from the system. This has to do with free iron, and as we already spoke about a few times, one of the important functions of frataxin is allowing the incorporation of iron into iron-sulfur clusters, which then go on to do all kinds of things in the mitochondria, very complicated pathways and such. So when the iron's not incorporated into these iron-sulfur clusters, it's free to interact with other substances inside of the cell. And that interaction is driven by an enzyme called 15-lipoxygenase. And the end product of that interaction is this ferroptosis, the cell death pathway. So basically, summing that all up in one simple sort of flow sentence is that an increase in free iron in the presence of 15-lipoxygenase causes ferroptosis and cell death. So that's just something I wanted to introduce. We'll go back to that in about another slide or so, but I wanted to put that out there. So as I said, we have a commitment to Friedreich ataxia, and we've actually been studying PTC 743 for about a decade in this disease state. So we have preclinical data that demonstrates the mechanism of action of this drug, how it works. And I'll show you some information showing both cell protection and then also an increase in frataxin mRNA levels. We have completed a phase 2 study. The last patient in that study was in 2014, so it was completed quite a while ago, that showed a benefit in Friedreich's ataxia patients up to 24 months after treatment. And then we have a robust history of safety in both pediatric and adult patients in diseases outside of Friedreich's ataxia and also from that phase 2 study. So I'll spend a little bit of time talking about that as well. So as I said, the main enzyme that's important for that ferroptosis cell death pathway is called 15-lipoxygenase. And what we've shown is that vatiquinone, or PTC 743, has a really strong effect on decreasing the activity of 15-lipoxygenase. You can see that in that nice curve. At very low concentrations, we see a decrease in that 15-lipoxygenase enzyme's activity. And then in a test system, we've shown that patient cells are able to survive longer in the presence of vatiquinone than they would have otherwise. This is data that came out of Rome in 2019. It was a really interesting study looking at frataxin mRNA levels. As was already discussed, mRNA is the instructions that allows the frataxin protein to be produced. And on the far left-hand side of the slide, you can see the levels in healthy controls are about 1, is how they're graded here. And then in patients, the levels are much lower. And so that relates to the lower levels of mRNA levels. The lower mRNA levels relate to lower frataxin protein levels. And what was shown by this group in Rome is that vatiquinone, which is with the little green arrow there, restores the levels of frataxin mRNA to approximately control levels. So that's another important aspect of the mechanism of this drug. I mentioned we had a phase 2 study that completed a while ago. I wanted to share this important piece of data from that. This was an analysis done that compared patients who had received vatiquinone for 2 years to those in the FA-COMS study, the natural history study. So sort of first and foremost, thank you to everyone who participates in that study, because without your participation, we wouldn't be able to do analyses like this to show long-term benefit of our drugs. So we matched patients from our study to the FA-COMS study by their age, their gender, and their disease state. And what we showed was that patients who were untreated as per natural history would have had approximately 4.8-point worsening on the FARS over that 2-year time period, whereas the patients who were treated with vatiquinone over the same time period had a 1.8-point improvement. And so this is a good difference and led to us bringing the compound forward into the current trial, which is a phase 3 pivotal study. And then before we get on to the phase 3 study, I just wanted to highlight the safety of this drug. As I said, it's been studied for more than a decade, both in Friedreich's ataxia and in other diseases that affect the mitochondria, things like Pearson syndrome, Lee syndrome, mitochondrial epilepsies. We've administered this drug to more than 513 patients as of this data cut, which was December of 2019. Most of these patients were pediatric, and their longest exposure was more than 9 years. And so we have a demonstrated safety and tolerability of this compound. So this is our phase 3 study. It's a 72-week trial with a double-blind placebo-controlled phase and then a 24-week open-label extension. What I wanted to highlight in this is that our primary analysis is in subjects aged 7 to 21 years. And this is a fairly robust study. We've got about 126 subjects to be enrolled. We did start enrollment in January of 2021, and I'm very proud to say that our enrollment is on track despite the pandemic, and that is due to all of you, especially FARA's support and the researchers and also the patients, obviously. So thank you so much for enabling us to reach our goals on enrollment in this study. Our double-blind phase will be completed in early 2023. We're really excited about that. And again, this is a global study. We have 14 sites across North America, South America, Europe, New Zealand, and Australia. So that is it. Thank you.
Thank you, Tracy. Shayna, Dr. McCormick's coming back to tell us about some research she has.
Wonderful. Thanks so much for allowing me to come back. I'm going to share a couple of trials that we're working on at the moment. And I'll mention that this presentation is on behalf of the cardiometabolic core, so with Dr. Kim Lin, who you've heard from as well, who we work with very closely. So in Friedrich's, Kim has found, and Kyle, thank you for demonstrating here.
Those abs.
Look at that. That's awesome. Overall, on average, not in a particular given individual that's shown there, but cardiovascular fitness is lower than we might like. And fitness, which is indicated by VO2 max, or the amount of oxygen that the body can use, can be an outcome that may be important in clinical trials. I'm mentioning the symposium in particular because this gathering has been really critical to me, I know, in our research program in pursuing research questions. Folks asked us a lot of questions about the value of exercise and what the role for exercise training could be. And also they shared that the way that exercise is done needs to be modified based on where they are in the course of their condition. And so as a result, we started thinking about the role for exercise training. We also were thinking about molecules that may mimic the effect of exercise because we know that it has so many benefits. So in particular, we're studying NAD precursors. NAD stands for nicotinamide adenine dinucleotide. We know that in healthy individuals, exercise training increases the amount of NAD in the muscle. And that's in part because NAD is an important cofactor in mitochondrial energy production. So Jen showed you all a diagram of the importance of frataxin in mitochondrial energy production. And if you zoom down in even more deeply than what she showed you, you'd see NAD there. There are different forms of precursors for NAD that we get in our diet. NAD is shown at the top right there. And so there's been a lot of interest in different ways to boost NAD. My collaborator and colleague Joe Bauer from Penn has shown in animal models that NAD precursors, some of which are shown, can recapitulate and add to the benefits of exercise training. So the potential to boost the benefit we get from exercise is really an exciting one. Also, Matt Hershey from Duke has shown that an NAD precursor has improved the course of Friedrich-related cardiomyopathy in an animal model. So we had, at the time we were thinking about studies related to this, there was some human data with modest efficacy in other conditions, not Friedrich's, but overall reassuring safety. So that was a signal perhaps we could continue. The diagram on the right shows the synthetic pathway for NAD. We aren't going to go over it in detail except to mention its complexity, and it's one of the questions that we ask ourselves, what's the right way to boost NAD if that's indeed our interest? So I mentioned some of the initial studies that were done were not as exciting in individuals with overweight or obesity in whom we were trying to target, our colleagues were trying to target blood sugars. There's a few studies, though, I want to point out. One safety study was done with a high-dose form of NAD precursor called nicotinamide, higher than doses that we might typically think folks need, and were overall safe, with nausea being the main side effect that was reversible. In a different form of mitochondrial myopathy, one NAD precursor called niacin was found to increase muscle strength in comparison to controls. And more recently, in a study of otherwise healthy, overweight, and obese women after menopause, a different NAD precursor improved insulin sensitivity, which we would think would decrease risk of diabetes, and promoted more healthful muscle remodeling. So on the basis of all of those exciting data, we designed a clinical trial that we were really excited was funded by the National Institutes of Health. So for folks who are aware, the NIH is the government's main research body. So our taxpayer dollars at work in the service of answering this question in Friedreich ataxia. So our goal in this study is to measure the effect of an NAD precursor, a supplement called nicotinamide riboside, and exercise on fitness, so how well the body can take up oxygen during a test, and on blood sugar in individuals with Friedreich ataxia. It's a randomized placebo-controlled trial. Our goal is 72 individuals to achieve complete data on 64, ages 10 to 40. And so far, we have 19 enrolled in pandemic era recruitment, which we really appreciate because that's a lot to do. It requires 2 in-person visits at the beginning, at the end of the study. The intervention itself is 12 weeks. So we tried to design the shortest possible study we could to permit folks to fit it in between all of these other opportunities that are available to them. Eligible individuals are assigned randomly to one of 4 groups. It's what's called a factorial design. So everyone gets either in-home exercise training or an attention control, meaning some weekly check-ins. And they also get either nicotinamide riboside, the supplement, or a placebo. And I'll mention many folks enroll in the study because they're interested in the in-home exercise intervention. For folks who are not randomized to that group, we offer that same intervention at the end of the study. So anyone who is interested in pursuing it, if they choose, they can. We were grateful, I think, but at the time when we designed this study was pre-pandemic, to make the exercise in-home instead of supervised in person. And that's been the aspect of the study that translates to the pandemic era the most. Our coordinator team, I'll share this slide with Jen so she can post it or otherwise make it available, has some answers to common questions. How long is the study? 12 weeks. Do you pay for travel reimbursement with the companion? We do, up to an amount. It's okay if you don't exercise now. The form of exercise that we use is the recumbent trike, so there is that capacity consideration. And certainly folks can continue their usual exercise or PT during the study if they wish. The recumbent trike is yours to keep at the end. This is open to pediatric participants, and so the candy there is not dietary advice. It's instead for people for whom pill swallowing is a consideration to show how big the supplement is, because that is a consideration for some of our pediatric and even our adult participants. So that's one study in NAD precursors that I hope you all will find interesting. Another is based on that observation I shared with you of Matt Hershey's, that the NAD precursor may improve Friedreich-related cardiomyopathy in an animal model. So we're interested in following up on that as well. And in order to do that, to focus on the heart, we wanted to think about techniques to look at energy production in the heart itself, which is not trivial to do, but there is an MRI-based technique called 31 phosphorus magnetic resonance spectroscopy. That's a mouthful, but what that means is that we can use the MRI magnet to measure the relative concentrations of metabolites that have phosphorus in them. And one of those is ATP. You remember from your biology classes, that's the energy currency of the cell. And phosphocreatine, which is essentially a storage form of ATP. And the ratio that you'll see an example spectrum on the right, the ratio of phosphocreatine to ATP reflects in general how much energy is available in the cell. And that's been shown previously, as that panel on the bottom left indicates, to be decreased in individuals with Friedrich's. So that's one insight. We have a technique available to look at bioenergetic poise. We also have a technique available from our colleagues at Penn to measure how much NAD is actually in the muscle. Now here, this is data from skeletal muscle. We don't yet have a technique that works in heart, but we're working on that, where if you can see on the left panel, those little protons, H2, H6, and H4, come from NAD, and the size of those peaks are higher in a control individual as compared to an individual with Friedrich. So we're wondering if some of the interventions that we're designing to try to boost NAD could be reflected in this spectroscopy. So the application is another trial. This is a pilot study. We're going to enroll between 6 and 10 individuals and use a different kind of NAD precursor. We're most interested in safety in this pilot study, but we're also going to use the 2 MRI techniques that I mentioned to look at the energy availability in the heart and to look at the NAD availability in the muscle. The main questions we get about this: how long is the study? 2 weeks. Also travel reimbursement. No need to exercise for this one, but you do obviously need to be able to have MRIs as the outcomes show. So I'll make these slides available because they include contact information. And thank you again to everyone who's volunteered for ours and all other of these studies. We really appreciate it.
Thank you, Shana. And Dr. Lynch, you're up next.
Okay. And now for something a little bit different. So are we talking about a study that actually has 2 names? The first is a pilot investigator-initiated study about the safety, tolerability, and efficacy of olaparib and the treatment of advanced symptoms of FRDA, which for obvious reasons, we have shortened that title. Elamipritide for visual function in FA, or for short, Elvis in FA. So we'll just call this the Elvis study from now on. What is this? I don't think anyone has heard much about it. Elamipritide is a well-known small molecule that stabilizes cardiolipin, that is, it changes its turnover in the membrane. Cardiolipin is an unusual lipid which is a central part of the mitochondrial membrane, particularly in heart, thus the name cardiolipin. When you give elamipritide to cells in a dish from a variety of mitochondrial disorders, it increases ATP production, reduces oxidative stress, and improves mitochondrial respiration. One thing which has been found in multiple labs is it also raises frataxin levels in cell culture in FA mice, an effect we have replicated in some situations in our lab, and is really sort of the effect that I was expecting to go away when we tried to replicate it. But it in fact does do that. It's not huge, it's about 2-fold. In clinical trials, it's been in clinical trials in various formulations for the past probably almost 15 years. It barely missed its endpoint in Letter's hereditary optic neuropathy, for which, for those people who do not know, is a very strange mitochondria-based optic neuropathy where one eye gets visual dysfunction and then the subsequent one. So it gets very difficult to design the controls. It's another genetic mitochondrial disease. It has also shown apparent benefit in the cardiomyopathy of Barr syndrome, another uncommon mitochondrial disease whose major feature is cardiomyopathy. It's actually a mutation in the genes which make cardiolipin in the body. That disease has a hypertrophic cardiomyopathy, which is followed later by loss of systolic function, resembling another disease we might all know, Friedreich ataxia. It's another nuclear encoded mitochondrial disease, and this drug may reach approval from stealth pharmaceuticals and for Barr syndrome at some point here. Their data are actually quite good. It makes some sense then, and it might have applicability to FA. Switching gears, let's talk about vision in Friedreich ataxia. It's effective in a large number of individuals with FA. At any given moment, it may not be a large part of people, but serially over time, greater than 20% of individuals will have clinically meaningful visual loss. It is a modest acuity loss in most individuals, going from normal, which is 20/12, out to 20/40, 20/50, 20/70. Remember, the level at which you're able to drive is 20/50 in most states. It is also unusual with what you lose most is not your central vision, but your peripheral fields. You are unable to see what is next to you. You're unable to see, but you're able to see things in front of you. You also lose what's called contrast sensitivity, which I'm sure those of you who come to our clinic know very well. The world is not looking at black letters on a white background. If it were, it would be quite boring. It's looking at gray letters on a white background, or looking at colors on a background, which are lost before your typical visual acuity. What this means biologically is a particular cell within the retina called the retinal ganglion cell. It's the output cell for the retina, and it transmits the impulse to the central nervous system. That is the main thing, along with the optic nerve, which arises from the retinal ganglion cells, which is lost in people with FA over time. So who is at risk for clinically meaningful visual loss? People who have long GA repeat lengths or point mutations, and people with early age of onset as they get to their 30s, roughly speaking. So typically it's a person with a repeat length greater than 700 or a point mutation, and we're meeting them for significant visual loss as they go from 25 out to 70. So it's a different group of individuals than what you're hearing are the ideal candidates for the other clinical trials that are being talked about. So it obviously affects quality of life. I think many of you have seen this statement from the mother of these two individuals who I think she's watching since she asked a question earlier, that says something which I think everyone knows: vision is important. You know, they sent me to medical school to learn things like that. As you know from having come to see us, we have a vision chart which can measure anyone and watch it change over time. Early on, it's low contrast, gray letters on a white background, but then as you get to the 25 to 30 time, it becomes black letters on a white background. We can measure it at any point. You could also take a picture of the retina with what's called optical coherence tomography, which is a non-invasive way they shine a red light through your pupil, and you can measure the thickness, again, I don't have a pointer, of the neurofiber layer, which is what we watch go away in FA at the rate of 1 micron per year. I would note the reproducibility of an OCT is about 1 micron per year. So you would see clinically meaningful protection in about 2 years if you look at an OCT. We have done that in a limited number of people, and yes, you see it does go down. It goes down about 1 to 2 microns per year. So the rationale for doing a trial for assessing vision loss: we can quantitate vision very well. We count the number of letters you read on a vision chart, which sounds very simple, but it is very meaningful. We understand what's going on. There is very little aspect of central nervous system, which I should say brain visual dysfunction in FA. It's all at the level of the retinal ganglion cell. I remind you that that's part of the central nervous system, in fact. So we understood it. It's important. No one should debate that vision is an important thing to preserve. And there are a relatively large number of relevant patients who are in that 20% of individuals right now who have clinically meaningful changes in their visual acuity. The other catch is something which is somewhat coincidental. I remember Kim mentioned that people who have long GA repeat lengths are the ones who are most likely to get significant cardiomyopathy, though not limited to such individuals. So the people who have clinically meaningful vision loss are those who are likely to have clinically meaningful cardiac loss, in a range where it would be ideal to try and find a treatment for that. So it's, again, long GAH, longer GA repeat lengths, longer duration. So we have basically proposed a study. This is an investigator-initiated study. The sponsor investigator is the Children's Hospital of Philadelphia and myself. It is being funded by a grant from a large, well-known nonprofit entity called the Friedrich Ataxia Research Alliance. Stealth Pharmaceuticals, who produces a drug, is donating drug, but they're advising us, but they do not have a direct monetary support for this on the other way. It is not fully IRB approved at this moment, so I cannot tell you all the details, but I can show you a rough timeline. You see 2021s and 2022s here, so I think we're in 2021, aren't we? So you can get a rough idea of things. There are 2 doses of amiprotide. Needless to say, we'll be looking at people who are clinically affected, so they will have significant vision loss. We will obviously be assessing their cardiac function as they go along, so I'm not telling you anything confidential. Amiprotide comes in a variety of preparations. They're all administered by, in this case, subcutaneous administration. And you will have to come to CHOP a couple times. It's not the worst place in the world to visit. And you will hear in the upcoming days, weeks, when we announce enrollment and invite your participation. Thank you. And no one will be required to sing Elvis. And I will be forbidden from singing.
Thanks, Steve. Next up, Kara Fink from Riata.
All right, hi everybody. I'm Kara Fick, and I have the coveted position of being right before lunch, so thank you for sticking with us throughout the day. Hope you can stick through a little bit longer with us, but I'm going to tell you a bit about what Riata has been up to with Friedrich's ataxia. Let's see. Yes, got it. Like everyone else, I have these long legal slides, forward-looking statements, and some disclaimers. There actually are 2 disclaimers I do want to point out that are important, though. Today, I'll be talking about omavaloxolone, which is Riata's drug, and omavaloxolone is an investigational drug. Safety and efficacy have not been established by any agency, including the FDA. The second one is more of a personal disclaimer. I'm not a scientist, I'm not a doctor, like most of the panel has been this morning. But my secondary title at Riata is the FA historian. I've been around for a long time with you guys. So today, I'm gonna dabble into the science a little bit, but do a little bit more focus on where we've been, what we're doing now, and what we're hoping to do next. I have a couple of PhD colleagues in the audience if you guys have some scientific questions, and I might be able to lean on Dr. Lynch a little bit for some other things, but just wanted to put that disclaimer out there. So let's get into it. This is the FARA pipeline that you all have grown to know and love over the years, and I wanted to point out where Riata falls on this pipeline of therapeutic approaches. So we're in this red section up here that targets improving mitochondrial function and reducing oxidative stress. Now, this is the little bit of science that I'm going to get into. The rationale for using omavaloxolone in Friedreich's ataxia. So in the cells in our body, there are all sorts of processes going on all the time. And a lot of these processes are controlled by transcription factors, which basically tell your cells what to do and when to do it. So there's a transcription factor called NRF2, and it does a lot of things, but it coordinates processes that mitigate inflammation, reduce oxidative stress, and help to restore mitochondrial activity. Now, it's normal for this to turn up and turn down and turn on and turn off, but the problem is that in FA patients, NRF2 signaling is significantly impaired. And fun fact, the reason that we know this is because of a research grant that FARA provided to an academic institution a while back. So those funds that everybody raised at Ride Ataxia yesterday goes towards things like this. Now, this is where OMAV comes in. OMAV, short for omavaloxolone, is an NRF2 activator. So that's why we think it may be potential as a therapeutic agent in Friedreich's ataxia. Now, over the years, there have been 5 major components to Riata's work in Friedreich's ataxia. There's been the MOXIE Part 1 study, which was really looking at safety and finding the right dose. The MOXIE Part 2 study, which was looking primarily at safety and efficacy, specifically of a 150 milligram dose. And then anybody that completed Part 1 or Part 2 could roll into an open label extension. So Part 1 and Part 2 were placebo controlled. Some people got drugs, some people got placebo. But in the open label extension, everyone receives OMAV for the long term. Now, the data that came from Part 1, Part 2, and the open label extension was used to conduct the baseline controlled study and the delayed start analysis. Now, I'm not going to get into results of any of these because, like I said, I'm not a scientist, And FARA did a really good job of giving a talk on a webinar recently that you guys can go to see to look at the results. But these are all of the components that have been involved in Reata's involvement in Friedrich's ataxia. Jen made a mention of how much effort patients have put into participating in clinical trials over the last year and a half in particular during the pandemic, and I really want to emphasize how important that has been for the participation, especially in the open label extension, and the patients who have continued to go to their visits and keep up with the study protocol during the open label extension period, because that has been what has enabled us to do the baseline controlled study and the delayed start analysis. So this is where we get into the history. Let's see, this might help. Sorry to you guys on that side of the room. I know you've been neglected with the laser pointer, but sorry. So this is a timeline of the last 7 or so years of Reata's involvement in Friedrich's ataxia. And I want to point out a couple of things on here that are in red that have been driven by FARA that are really important to highlight along the way. So back in 2009, paper was published that talked about NRF2 and its role in Friedrich's ataxia. And that was the whole rationale that gave Reata reason to get into this space. So in 2014, we started part 1 of the MOXIE study, enrolled our first patient a couple months later, and then about 2 years down the road, we finished and had data presented from part 1. So that's here in 2017. Shortly afterwards, FARA hosted a really, really important meeting called the Externally Led Patient-Focused Drug Development Meeting, ELPFDD. And basically, this meeting was to allow FA patients to tell the FDA what they want from future treatments in general. Nothing specific, but in general, what do they want from future treatments? Reata then started part 2 of MOXIE and enrolled the first patient. And a lot of us may have been in this room together 2 years ago when we announced data from part 2 and the fact that we had met our primary endpoint of that trial. Now, as we all know, the COVID-19 pandemic hit shortly afterwards. So I'm going to start to get now into where we are today. So back in August of 2020, we met with the FDA with our results from MOXIE Part 2, and we shared them with them, but the FDA told us, we're not concerned about your endpoint, but we're just not sure that this is enough evidence for us to be able to move forward at this point. So we said, okay, let's do some more. So then we went back, we did the baseline controlled study, we met again with the FDA, and they said, this is great, just still not sure that this is enough evidence to keep moving forward. That leads us to this very important initiative that was driven by FARA earlier this year. This is the FA Community Petition that had about 75,000 signatures. Pretty incredible when you think about that. Now, it may sound a little weird that I'm bringing this up because this petition was actually submitted to Reata and to the FDA. But it essentially was urging Reata to submit a new drug application on an urgent basis and asking the FDA to consider approving that application based off of the evidence that was already available. So after that petition came around, we did another analysis, the delayed start analysis. We presented that to the FDA, and this time they changed their minds. They said, okay, you know what? Go ahead and submit a request to us to meet for a pre-NDA meeting. And this is a meeting where you get together, you talk about what it's really going to take to submit an application to the FDA and work out any of the mechanics of that. So today, we've completed that meeting. We just put out a press release maybe a week or so ago. That meeting has been completed, and we plan to move forward with submitting an application to the FDA in the first quarter of 2022. Now, the question I get from everyone is, what's next? And that can be a little bit hard to describe. So I'm going to do my best here, but luckily I saw on the agenda there's an FDA 101 talk after lunch, so that may be able to fill in a few of the blanks. But we plan to submit our NDA in the first quarter of 2022. Then the FDA will have some amount of time to decide whether or not they're going to accept or reject that application. If they accept it, then they get some time to review it and make their decision. And ultimately, the FDA will decide whether they are going to approve or reject that application. So that's where we're headed. I hope that has helped to give some updates on where we are now. Like I said, if there are questions about some of the data or the science, I can certainly connect you guys with the people who can give you those answers, but appreciate the time to be able to update you here. Thank you, guys.
All right, well, thank you to all of our panelists. We have about 15 minutes for questions from all of you. And Felicia, do you have any questions from online you want to start us off with? Not yet? Okay. Kyle's got a question. Ron?
Kyle Bryan? Thank you. For the PTC study, what, if at all, are the differences between what was done when it was EPI and now that it's PTC? And do we expect the same, or are we hoping for a different result?
So that's a great question. So as Kyle brought up, we, as PTC, actually acquired a smaller company called BioElectron that had done the initial studies and the phase 2 study that I shared some of the information from. That was a long time ago that they did that study. The last patient last visit was in, I think it was April of 2014, September 2014, if memory serves. So things have changed in our understanding of what is needed for a study that is successful in Friedreich's ataxia. And what I mean by that is, when you do a clinical study, you're limited by like what you see in a certain period of time. So a trial can only be a certain length of time. That first study was only 6 months. So we've learned since that time that 6 months isn't enough time to see a change in a Friedreich's ataxia patient, whether or not the drug works. So that's one of the reasons why we extended the length of this study out to 72 weeks instead of just 6 months. The other thing is the endpoint that's used. In the initial trial, it was the FARS was one of the exploratory or secondary endpoints. And in the new study, we're using the mFARS. And the mFARS has been shown to be more psychometrically valid, meaning that you see the changes in a patient more appropriately than you would see in the FARS, which includes some other things that don't really tend to change as much over the time course that you have in a clinical trial. So we've learned from our own studies and subsequent studies, and hopefully have designed a trial that will demonstrate the benefit of this drug.
Thank you.
Do we have more questions? Wow, everybody's ready for lunch. Like, Tim? Microphone's coming.
In looking at the trials, and in particular, the gene therapy trials, to the degree that they are one shot, one try processes, would there be any general guidance, insights that could be offered to decision makers as to the wisdom of being first in line versus standing back and waiting if truly the opportunity is a one shot per se chance? If that question makes sense or shows my naivety.
Tim, thanks for that question. I was actually thinking of something along the same lines, which is, I think the question is related to decision-making in terms of clinical trials. And we have some materials on the FARA website, some new materials specifically related to gene therapy trials, and that try and help you go through that decision-making process and thinking about the risks and the potential benefits, and kind of how to weigh, a process for weighing those out for you, because for each and every individual, whether it's a gene therapy trial or it's a small molecule trial, you should go through this process of considering risks and benefits and what's being asked. And for each person, it's very individual. And so, we want to support you in being able to go through that process. And so there are materials on the website that can help you do that. But it really, deciding to participate in a clinical trial should be something that you consider carefully and really think about. And you don't have to do that alone. You have medical professionals to help you do that as well. So the physicians at your clinical research network sites can be a source for you in terms of sitting down with somebody and kind of going through that information and trying to, come to that decision, I think is, one important resource. But these are difficult decisions to make and opportunities to evaluate. And for each person, there isn't a correct or an incorrect answer. I don't know if Dave or Shana, do you have additional thoughts to add to that?
Well, I did notice that Tim's first question was a yes or no question.
Sorry.
But no, you have to weigh your own personal, where you are in life, what you're doing, what the trial involves, both risk, benefits, and how much it interferes with your life, and how much, in some people, participation in trials is part of their therapy, and other people, it's not. That's fine. And think about whether one wishes to do it, because as you do come with multiple trials at your discretion, there is always the phrase in the consent form that you have the right to drop at any time. That said, I would hope that people wouldn't drop just for the sake of dropping because they saw something newer that might be around there, because then you end up perhaps messing up might end up multiple studies, because dropouts are a big issue with things. Because the way we analyze data by intent to treat, if there are lots of dropouts, gets very confusing.
Yeah.
Randy Ipe again. I was wondering if I could follow up to the question I asked the earlier panel about plans among this panel for pediatric opportunities in some of the clinical trials, given that this is a disease measured day by day in neurodegenerative progression and the strong pediatric component of our community.
Nancy, you want to go first?
So, yeah, so we are planning to study patients as low as we can possibly get them. And I think, David, you mentioned that it's rare to get patients less than 5 years of age.
Yeah.
So we will go as low as we possibly can and study these patients. We're talking to the FDA, we've talked to the European agencies, and we've agreed with them that we will do studies in pediatrics, not just in adults.
So as I mentioned, in our trial, we have subjects enrolled down to the age of 7. That's supported by the robust safety and tolerability data across other disease states where we've treated subjects as young as 1. To that point, it's difficult to enroll anyone in FA less than 5 to 7 years old, but we're going as low as we can in our trial.
Kara, maybe, do you want to speak to where things are with, I think some of the other trials do include kids, but I know the Riata study and the Retrotope study, Retrotope went down to, I think, the age of 12, but
Right.
16.
So Riata's clinical trial enrolled ages 16 to 40. We went down to 16. The trial has been completed, so we're not enrolling any new patients. As we move forward with our regulatory process and we're starting to prepare our European regulatory submissions, we are planning for a pediatric clinical trial in Europe, because that's part of what you have to do for submitting in Europe. Now, here in the US, that's a little bit more complicated to answer. Perhaps one of our primary investigators may be able to chime in on that.
And so,
Dave, just make sure you're using a microphone.
Oh, okay. I thought people could hear me. Just, I can yell. So, speaking in a very broad sense, relevant to everyone here and everyone in the gene therapy and design and Execure, there is this word called dose. That is to say, until you dose a 12-year-old, you do not know that the dose is the same as a 16-year-old. I will now pick out one very specific finding from Moxie Part 1, which is that there is I compliment Riata, yes, I was the PI, but I'm complimenting them, on doing a detailed dose response curve. And when you go from 160 milligrams a day to 300, the effect goes away. So there's a twofold difference between optimal effect and no effect. So now go outside of your original age group and what's the dose for a 12-year-old. You actually theoretically really have to know that in order to know how to design that study. You might get that from animal studies, you might get that from limited human studies, but it's not something, it's not even, so there's a safety issue, but there's also the efficacy issue. If you give the wrong dose, you could get rid of your effect. Welcome to some of the tough things that have to be done.
So I got a question, because my background is in depression and suicide research at New York State Psychiatric Institute. And I'm noticing the lack of mental health kind of research that we're doing. We're doing a lot of biomechanics, and I think that's awesome. But for the exercise program, for example, to have a psychiatrist on board to do a Hamilton Beck or something like that, which I think exercise, in my limited ability, is a really positive benefit for mental health stuff. And I think mental health is a big issue, not only with caregivers, but with the subject, patients. Can you address that?
So I appreciate that question, and I think you are highlighting a need for us to more explicitly recruit our colleagues from the mental behavioral health side. Since you asked about how we address it in our study, we do in a few ways, though not perhaps to the extent we could. So one way in which we address it from a safety standpoint, we have a standardized kind of system-based assessment to monitor people's safety, and one of those dimensions includes mental psychiatric health. So we are gathering at least where folks start and what their impressions are. The other thing I'll mention that Kim has been working very hard on is incorporating a new FA-related health quality of life scale that does incorporate questions related to mental health, which I'd be really curious to see how exercise affects that. The other just thing to mention is, the more we, of course, include, the more substantial the burden on participants. So we really want to make sure we're thinking of the right questions and the right way to focus people. But your point is incredibly well taken, and so I'm hopeful that this new scale will capture some of those important dimensions that you mentioned.
Great, thanks.
Alicia, we have some online questions?
I've got at least two.
Two, okay.
Several people asked about EMA status for Reata in Europe, and also is there are plans for other countries, other approvals?
So we are still planning to move forward with an EMA submission. There hasn't been a public statement on the timing around that. And unfortunately, I don't have any further information that I can provide on some of the other countries' submissions. But if you continue to watch Reata's social channels and our press releases, that's the best way to get the most up-to-date information, since I can't give you a fully satisfactory answer today.
Felicia, you have another?
Yeah, the other question is about gene therapy approaches for the eye to treat vision loss. If Dr. Lynch could comment more on that.
Okay, so it's not exactly about Elvis, but I had to say Elvis just because I like singing. In my opinion, and I'm now expressing my opinion, I'm not commenting on any of the other companies' platforms, in all reconstitution of frataxin, it's real estate. Where you get to and where you are is the important thing. The eye is in some ways a very attractive target. It would make sense if one of the limitations of gene therapy or any of these other therapies is where you get to. The eye is not a hard place to get to. It's a little squeamish, you just inject in it, but you can get there. And it would seem rational that someone would want to develop a gene therapy, frataxin-based gene therapy for the eye. I would hope that some people are doing that. I cannot share anything more at this point.
Maybe I can add one or two
You can do
things.
that, so I can add.
So
yeah.
FARA's had conversations with a few different gene therapy companies to talk about, targeting the eye for a gene therapy trial. And one of the challenges was that there wasn't a good model to do some of the preclinical work. And so we were able to identify some researchers at the University of Florida who have expertise in studying vision loss and inherited diseases, and they've actually been able to make an FA mouse model that mimics the vision loss that we see in FA in the mouse. So we now have a model where we can test a proof of concept, and they can do some of the work they need to do to understand delivery and dose and things like that. And Barbara Tate, who's here today, who's new on the FARA staff over the last 18 months. So this is her first FA symposium. Barbara's our chief scientific officer. She has been working hard to reach out to a lot of different companies who have gene therapy programs in the eye to talk to them about feasibility. So as Dave said, no promises, but we agree with you that this is an unmet need and an opportunity to maybe think about targeting the eye for future therapies, and hope to have new partners in that space soon. Yeah, I think we have one more question.
So can I ask it?
You can, Deborah, absolutely.
So Dave, got a question for you about your eye study. Is diabetes an exclusionary criteria because of the complications associated with the diabetes and the eye?
So I cannot.
And you know who I'm referring to, my son has.
Yeah, so I cannot comment, since it has not been fully IRB approved, about the specific inclusion criteria. But I think, you know, we will be, based on my opinions, which are pretty well known, that we will be looking to be as inclusive as is rational, and we will be scientifically based. We would not want people participating in this study whose vision loss is from macular degeneration. We would not want people participating in the study whose vision loss is from refractive error, that is new glasses, or cataracts. We would be wanting people whose vision loss is FA related. You could imagine that if I were writing these inclusion criteria, I would probably be sure that a person does not have diabetic related vision loss, and we could successfully measure the FA component. But that was, I used the word "were," indicating that it's a contrary to fact condition, perhaps, because I used the subjunctive tense.
So I look forward to hearing from you.
So I violated no rules with what I just said.
Well, thank you for our panel, for giving us updates on all these exciting clinical programs, and to you for your questions. 2 announcements before you go to lunch. One is that there is interest in having a photo taken. So all of our individuals with FA, if you'd go out to the lobby area for a photo, I have no doubt Kyle Bryant is leading this effort, and will be responsible for organizing the photo shoot out in the lobby. And then there are box lunches out in the reception area here. You can bring your lunch back here, but we also have another ballroom, the Independence Ballroom. And if you go back through the main lobby of the hotel and to the right, you'll find the Independence Ballroom. And that gives you another location where, you know, you can have a slight change of scenery and enjoy your lunch, and be back here at 1:15. Thank you.